Welcome to this video. This botany class is for plus 2 students. I wish to give you some previous year's questions and explanation of the same content. There are two questions given. They are, which among the following is female gametophyte? Four options given. They are nucellus, endosperm, embryosac and antipodals. What is the correct answer? Yes, embryosac is the correct answer. Then, what is the second question? Observe the diagram given below and answer the following. Then, this question consists of three sub-questions. First one is identify the structure. How many cells are seen inside the structure? What are the middle nuclei together called? See children, these two questions are from second chapter, that is sexual reproduction in flowering plants. Then in the case of the second question, to answer the sub-question A, it is very easy. The answer is embryosac. Why? Writing the answer of these two questions, those who have a thorough knowledge about the structure of embryosac only can attempt the answer. So we can study the structure of embryosac. Embryosac is otherwise known as female gametophyte. Then what do you mean by gametophyte? Gametophyte is a stage which produces sex cells. So here the female gametophyte means this gametophyte produces female sex cells. So it is very clear an embryosac consists of a female gamete. So what is the significance of embryosac and where is it located, etc. we have to know. Then a flower is known as the reproductive unit of a flowering plant. A flowering plant is otherwise known as angiosperm. A flower consists of two parts. They are reproductive part and non-reproductive part. The reproductive part consists of androecium and gynoecium. Androecium is the male reproductive part, while gynoecium is the female reproductive part. So this female gametophyte or embryosac is present in this gynoecium. So, a gynoecium consists of carpel. The number of carpels vary in different flowers. There may be monocarpellary, bicarpellary and multicarpellary. Monocarpellary means if there is only one carpel is present, it is said to be monocarpellary. If there are two carpels present, it is said to be bicarpellary, while there are many carpels present, it is known as multicarpellary. Similarly, if the carpels are united, it is said to be syncarpous. If the carpels are free, it is known as apocarpous. Apocarpous. Yes, next, what is the structure of a carpel? A carpel consists of mainly three parts. The solemn basal portion is known as ovary, and this portion is known as the stigma, and this is the style. Stigma is the landing platform for the pollen grains. Male gametes are present inside the pollen grain, while the style connects the stigma with the ovary. Inside the ovary we can see ovule. After fertilization, ovary gives rise to fruit and ovule gives rise to seed. Some of the cells which are present inside the ovule differentiated into megaspore mother cell. This megaspore mother cell undergoes reduction division. Reduction division is otherwise known as meiosis, and to produce 4 cells. It is reduction division, otherwise known as meiosis. What about the ploidy of the cells which are produced after meiosis? Yes, the nuclei are haploid. Then what do you mean by haploid? If there are only one set of chromosomes present in the nuclei, they are known as haploid. What is the other term? That is diploid. If the chromosomes are arranged in 2 sets, that is known as diploid. So after meiosis, all the cells which are formed in haploid condition. Then here, for out of these 4 cells, these 3 cells degenerated and they get destroyed, and there is only one nuclei functional. So this development of the embryo sac is taking place from this only one megaspore. So it is known as monosporic development of embryo sac. Monosporic development of embryo sac. Why it is known as monosporic embryo sac development? Because the embryo sac is developed from only one megaspore. Next, how this embryo sac is developed from this one megaspore? Let us see. This megaspore undergoes 3 repeated mitotic division. So, 3 repeated mitotic division. 1, 2 and 3. It is divided into 2. Then this is divided into 4. Here it is divided into 8. Here during reduction division or meiosis, the cells are haploid. Then here onwards all the divisions are mitosis. So what about the ploidy? The resultant structure or the resultant nuclei consists of the same number of chromosomes which are present in the parent nuclei. So here the ploidy is haploid. In the parent nuclei it is haploid, but here it is diploid. So here also it is haploid. Do you understand? Then how these nuclei are arranged in the embryo sac? Embryo sac is developed from this one functional megaspore. So how are they arranged? They are arranged in the embryo sac in such a way that 4 of the nuclei moves to opposite poles. In the next step or in the next stage, 1 nuclei from each pole move towards the center. So they are known as polar nuclei. Usually there are 2 poles present in the ovule. This part is known as chalazal end. Chalazal end, while this portion is known as the micropylar end. Micropylar end. The 3 nuclei which are present in the chalazal end are known as antipodals. Antipodals, and the three nuclei which are located in the micropylar end known as egg apparatus. Then inside this egg apparatus, middle nuclei which is present in the egg apparatus is known as the egg. Actually, egg is the female gamete. Female gamete. And the two nuclei which are present on either side of this egg is known as synergid. The arrangement of nuclei inside the embryo sac. Three nuclei, they are known as egg apparatus. They are present in the micropylar end. The three nuclei, they are known as the antipodals. They are present in the chalazal end. In the middle, there are two nuclei. They are named as polar nuclei. But later, they are differentiated into secondary nuclei. Secondary nuclei. So, the development of embryo sac was first studied in polygonam, so that it is known as polygonam type embryo sac development. What is it called? Polygonam type embryo sac development. Then, in the first question, that answer is embryo sac. What about the third and fourth sub-question of the second question? In this question, how many cells are present inside the embryo sac? Yes, can you say how many cells are present? 1, 2, 3, 4, 5, 6, 7, 8. But these are not cells. These are nuclei. This condition is known as 8 nucleate stage. Can you say how many cells are present? This is one cell. 1, 2, 3, 4, 5, 6. But these two together form, two nuclei together form one cell. So total 7 cells are there. 7-celled 8 nucleate stage. Then there is another question. Name the nuclei which are present in the middle. It is known as secondary nuclei or polar nuclei. Then what is the significance of this polar nuclei as well as this egg? Egg is the female gamete. As we all know, female gamete undergoes fertilization. What is fertilization? Fusion of male and female gamete is known as fertilization. And the resultant structure is zygote. Zygote develops into embryo. Embryo is the baby plant. Then baby plant develops to new plant. Then the secondary nuclei develops into endosperm. This endosperm is a nutritive tissue which nourishes the zygote, which nourishes the embryo. So that is the significance of the secondary nuclei. I think it is very clear about the structure of embryo sac. That's all about today's class. Thank you for watching this video.